I think I can start now.
Yes, sir, you can start.
Okay. Okay, I will start now. So we have 3 units left in this syllabus, chap unit 11, 12, and 13. I will share the screen now. Is the screen visible?
Not yet, sir, not yet.
Unit 11 is visible?
Yes, it's visible now, sir.
Okay, thank you. So unit 11 is pharmacognostical studies and herbal formulations. So the present unit deals with the study of adulterations in herbal drugs, methods for their evaluations, herbal formulations, and herbal drug interactions. Herbal medicine is the oldest form of healthcare known to mankind, used by all cultures throughout the history. It played a vital role in the development of modern civilization, and nowadays also it is doing a lot of help in the medicine. So after studying this unit, you should be able to describe drug or adulterations, their reasons and drawbacks, explain the drug evaluation, then list various drug formulations, and discuss herbal drug interactions and their mechanisms. So first, adulteration of drug. First, let us know what is drug. Drug may be defined as an article recognized in the official compendia, that example India Pharmacopoeia, National Formulary of India, intended for use in diagnosis, cure, mitigation, prevention, or treatment of diseases in humans or other animals, or intended to alter the bodily function or structure of humans or other animals. So, drugs can diagnose, cure, mitigate, prevent, or treat the diseases in our body. So, that's why it is called as drug. Any adulterated drug means the one which does not conform to the official requirements. Adulteration involves incorporation of impurities, spoilage, deterioration, admixture, sophistication, and substitution. Adulteration can be accidental or deliberated or intentional. The quality, value, or usefulness of the drug may be affected or destroyed by the action of microorganisms, insects, rodents, etc., due to improper storage of drug material. Factors such as temperature, moisture, and sunlight may also affect the proper storage. Now, types of deliberate adulteration. There are 5 types of deliberate adulteration, like confusion of common vernacular nomenclature by cross substitution with different plant material, substitution with artificially manufactured substances, substitution with exhausted drugs, and addition of foreign matter. So, depending upon the reason of adulteration, we can divide into 5 types of deliberate adulteration. Now, drug evaluation. Drug evaluation means confirmation of its quality and determination of its quality and purity and deterioration of nature of adulteration. Such an evaluation is carried out to know biochemical variation in a drug, deterioration due to treatment or storage, or substitution and adulteration. So, to evaluate the drug whether it is of proper quality, we have to give we have to do various methods to evaluate the drugs. So first one is morphological or organoleptic evaluation. The morphological or organoleptic evaluation refers to the evaluation of drugs by color, odor, taste, size, shape, and special features like touch or texture, etc. So morphological evaluation is mainly about size and shape and color and odor. Microscopic evaluation. In the identification of crude drugs, it is often necessary to examine the anatomical structure. This involves the microscopical examinations of sections of material. In some drugs, transverse and longitudinal sections are taken and arrangement of tissue is observed. Chemical evaluation. Determination of active constituents in a drug by chemical process is referred to as chemical evaluation. Thus, chemical evaluation can be used as method for the analysis of active constituents. It comprises of different chemical tests and chemical assays. So, different tests and chemical assays are required to test the chemical value of the drug. The isolation, purification, and identification of active constituents are chemical methods of evaluation. Then, physical evaluation. Evaluation of drugs on the basis of important physical properties of active constituents is known as physical evaluation. So, the various physical physical qualities like density, optical rotation, refractive index, melting point, specific gravity, and all these are discussed in physical evaluation. Then biological evaluation. The drugs which cannot be assayed satisfactorily by chemical or physical means are evaluated by biological methods. Thus, potency of crude drugs or its preparation is determined by means of its effect on living organism like bacteria, fungi, animal tissue, or entire animal, and it is called as bioassay. This method is done for conformity of therapeutic activity of raw materials or finished products. So we can do biological evaluation also, like the effects on living organisms like bacteria, fungi, animal tissue, or entire animal. This is called as bioassay. Then herbal drug formulations. World Health Organization currently encourages, recommends, and promotes traditional herbal remedies in national healthcare programs because such drugs are easily available at low cost. These are comparatively safe, and the people have faith in such remedies of herbal drug formulation. According to the study, 70 to 80% of people living in rural areas are dependent on herbal medicine for the treatment of day to day diseases. According to one estimate, more than 700 mono and polyherbal preparations in form of tincture, tablets, capsules for more than 100 plants are in clinical use nowadays. Ayurveda, Unani, and traditional Chinese medicines are important system of medicines largely based on medicinal plants. The general public's use of herbal drugs has increased over the past 10 years. So, herbal formulations are mainly under Ayurveda, Unani, and traditional Chinese medicine. Now, classification of herbal formulations, it can be divided into broadly two categories: herbal medicines and herbal cosmetics. The herbs are considered to be medicinally important if they possess the pharmacological activities of possible therapeutic use. The use of herb is known as naturopathic medicines, Unani, Siddha, homeopathic systems of medicine. So all these come under Ayush, that is Ayurveda, Unani, Yoga, and Siddha and homeopathic medicines. Now there are different types of herbal medicines and their uses are written here. Then herbal cosmetics. The substances specially prepared to enhance beauty and increase the attractiveness of a person are known as cosmetics. So cosmetics can be of different form according to dose and the part where it is used, like emulsions, powders, oils, jellies, suspensions, and different products depending upon the part of body on which it is used, like skin, hair, nails, teeth, these all. Then herbal drug interaction. Almost a century has passed and it has witnessed limitations of allopathic system of medicine. Lately, herbal medicine has gained momentum with the evidence that it has no side effects. Ayurveda, Unani, and homeopathy medicine are the important systems of medicine largely based on medicinal plants. However, the prevalence of clinically significant interactions between herbals and medication is unknown. A factor that may account for lack of data on the true prevalence of herbal drug interaction is that the following: a lack of information concerning the contents of the herbal products. So there is a lack of information about the contents of the herbal products. Also, incomplete or inaccurate product information and multiple ingredients. Because of these factors, we may lack the data about the prevalence of herbal drug interaction. Then, overview of drug interaction. Drug to drug interaction refers to an alteration of the effect of one drug caused by the presence of second drug. Drug nutrient interactions similarly refer to the alteration of the effect of drug or nutrient caused by the presence of second agent. Drug interaction may be beneficial or detrimental. Then, drug interaction. Drug drug interaction occurs when two or more drugs react with each other. Drug food beverage interactions results from drugs reacting with foods or beverages, and drug condition interactions may occur when an existing medical condition makes certain drugs potentially harmful. So there can be drug drug interaction, drug or food beverage interactions, or drug or condition interactions. Mechanisms of herbal drug reaction interaction. The interaction between pharmacologically active botanicals and drug involve the same pharmacokinetic and pharmacodynamic mechanisms as drug drug interaction. Pharmacokinetic interactions may involve alteration in absorption, distribution, metabolism, or excretion of the drug. Pharmacodynamic interaction, on the other hand, is named after the relationship between the drug concentration and the pharmacological response for a drug or botanical. So, pharmacokinetic means what the body does to the drug, that is, metabolism and excretion, absorption, distribution. These things are under pharmacokinetics, and pharmacodynamics means what the drug does to the body. So, it can have various pharmacological responses in the body, that is called as pharmacodynamics. Now, adverse drug reactions. In adverse drug reaction can be defined as an unexpected, diminished, or enhanced pharmacological activity of a drug or noxious response to a drug when used alone in humans at doses used for prophylaxis, diagnosis, or therapy. So, adverse drug reactions are classified as type A and B. Type A or augmented reactions are normal pharmacological effects of drug exaggerated to the point of being undesirable or intolerable for the patients. Example of drugs indicated type A in reactions include warfarin or heparin, which cause bruising. Then type B reactions have more severe adverse effects unrelated to unknown pharmacological action of a drug and include most immunological reactions. Example of a type B reaction is an anaphylactic reaction to penicillin. So, reactions which are normal pharmacological reactions in higher doses or undesirable doses is type A reaction, and if it is severe with severe unknown pharmacological action, then it is called as type B reaction. Then classification of drug interactions. There are three types of drug interactions: drug drug interactions, drug nutrient interactions, and herbal drug interactions. There are various drugs and adverse effects because of this drug. Some drugs are allopathic, some drugs are herbal, and different pharmacological actions and adverse effects are written here. Now we come to unit 12, evaluation and quality control of pharmaceuticals. The objectives of this unit is that understand various pharmacopoeias and their objectives, discuss the origin of pharmacopoeias, then differentiate between pharmacopoeia and formulary, differentiate between national and international pharmacopoeias, understand the pharmacopoeial monograph format, define quality control and quality assurance, understand the need of quality control of pharmaceuticals, understand the pharmacopoeia commission, understand the need of quality control of pharmaceuticals, and elaborate the various quality control tests for activities on drugs and preparations, their description, formulation, analytical composition, physical constants, main chemical properties used in identification, standards for strength, purity and dosage, chemical tests for determining identity and purity, and they are usually published under government jurisdiction, whereas formularies simply list drugs or collection of formulas for the compounding of medicinal preparations. However, sometimes the term pharmacopoeia and formulary are used interchangeably. So pharmacopoeia is a detailed description of all the drugs and their properties, whether physical or chemical, and formularies are simply the list of drugs and collection of formulas for the medicinal preparations. Each country has its separate sets of regulations pertaining to drugs and related substances, which are mentioned in the pharmacopoeia of the country. A few pharmacopoeias worth mentioning include Indian Pharmacopoeia, United States Pharmacopoeia and National Formulary, British Pharmacopoeia, European Pharmacopoeia, Chinese Pharmacopoeia, and Japanese Pharmacopoeia, and so on. Then, overview of Indian Pharmacopoeia. As per the Drugs and Cosmetics Act 1940, the Indian Pharmacopoeia is the legally recognized book of standards for the quality of drug substances and preparations included therein. The first Indian Pharmacopoeia was published in the year 1955. Then there is a term called Indian Pharmacopoeia Commission. The IPC is committed to the promotion of highest standards of drugs for use in prevention and treatment of diseases in human beings and animals, keeping in view the special features of the pharmaceutical industry in India. The overall objective of IPC has been the creation of a compilation of standards that reflect the state of the industry in the country and the production of testing capabilities of the units. Then Indian Pharmacopoeia monograph format. First, we have to write the description, means the physical properties of the drugs. Then identification tests, including infrared spectrometry and other tests. Non-specific purity tests, such as light absorption, optical rotation. Impurities, both related substances and impurities that may be present as a result of method of manufacture or from degradation on storage. Then impurities other than related substances that may require control, such as heavy metals, inorganic impurities. Other tests such as loss on drying or water, assay and limits for purity calculated on anhydrous, dried or solvent free basis as appropriate. To the extent possible for assay, a robust and precise, though not specific, method is given rather than specific stability indicating and complex one. In the case of non-specific assay methods, control of impurities is provided separately by means of specific impurity tests. Each monograph is designed to be considered as a whole and should provide a reliable basis for judging the quality of the drug substances. Then overview of United States Pharmacopoeia and National Formulary. The United States Pharmacopoeia and National Formulary is a book of public pharmacopoeial standards. It contains standards for medicines, dosage forms, drug substances, excipients, medical devices, and dietary supplements. It is the best known national pharmacopoeia in the world, authorized by the government of United States and published by non-government or non-profit organizations. Now, monographs and general chapters. The USPNF is a single volume combination of two official compendia, the United States Pharmacopoeia and National Formulary. Then official recognition. The US Federal Food, Drug and Cosmetics Act designate the USPNF as the official compendia for the drugs marketed in the United States. A USP monograph is given under the following headings: official title, descriptive information including graphic formula, molecular weight, chemical names, and chemical abstracts. Then assay content, tolerance limits, packaging and storage conditions, USP reference standards, identification methods and analytical tests, melting range, residual water limits and determination tests, residue on ignition, that is purity test, tests for determining chloride, sulfate and sulfide with limits, limit tests determining heavy metals and organic volatile impurities. So these are the monographs listed in the USP. Now, in the British Pharmacopoeia, the current edition of British Pharmacopoeia comprises 6 volumes, which contain nearly 3,000 monographs for drug substances, excipients, and formulated preparations, together with supporting general notices, appendices, and reference spectra used in the practice of medicines, all comprehensively indexed and cross-referenced for easy reference. So there are 6 units, 6 volumes in British Pharmacopoeia with 3,000 monographs for drugs. Other national pharmacopoeias are like European Union, Indian, and British Pharmacopoeia. All the pharmacopoeia which are issued under the authority of government and their instructions have the force of law in their respective territories are referred to as national pharmacopoeia. So there can be pharmacopoeias of different countries like China, Egypt, India, Japan, Russian Federation, United Kingdom, United States of America. Then international pharmacopoeia. Developing countries not having a national pharmacopoeia have adopted one or of the another country or countries, in some cases the international pharmacopoeia, which was put forward by World Health Organization in 1951 as a recommendation aimed at minimizing or eliminating variations among various national pharmacopoeial standards. The International Pharmacopoeia is published by the World Health Organization Geneva, by the WHO Expert Committee on Specifications of Pharmacological Preparations, with its latest version released in 2003 in English. Now, quality control of pharmaceuticals. The the concept of quality control started off with an understanding that endpoint quality testing of a product unit is insufficient to assure its quality. Rather, the quality is required to be assured at each step of the manufacturing process so that the quality is built into the product. Now, quality is the totality of features and characteristics of product or service which bear on its ability to satisfy stated or implied needs. The term quality control comprises of two words, quality and control. Control is meeting standards or processes through which we establish and meet standards. So quality control. Then we come to the next term, that is quality assurance. In the European Union guidelines, quality assurance is defined as a wide ranging concept that covers all matters which individually or collectively influence the quality of a product. Inside an organization, quality assurance provides a management tool. The quality assurance provides confidence for the customer, whether that is a pharmacist, doctor, or patient, in the quality of the drug being supplied. An important part of systematic actions is the availability of complete system of standard operating procedures. Hence, although there is a specific department within the company called quality assurance, the Achievement of quality assurance is not the duty of this single organizational unit alone, but is the responsibility of all staff members who can influence product quality in any way. So it is a responsibility of all the stakeholders regarding the quality control and quality assurance of the drug. Now we can check different pharmaceutical substances by various control tests like general appearance, weight uniformity, hardness, thickness, friability, means all the types of physical properties and different tests like uniformity of weight, drug content uniformity, disintegration, quality control tests with parenteral or injectable dosage forms, sterility test, leaker test, clarity test, pyrogen test, quality control test emulsions. We have to test whether one is dispersed phase or dispersion medium, determination of particle size and particle size distribution, determination of viscosity, determination of phase separation, determination of electrophoretic properties, and quality control of test suspensions like sedimentation volume, degree of flocculation, rheological consideration. Rheology is the study of flow of matter, mainly liquids, micrometric methods, then suppositories by visual examination, weight variation test, hardness of suppositories, that is the breaking test, melting range test, liquefaction time, quality control test of ointments like penetration, absorption of medicament into bloodstream, irritant effect. So these are some of the tests done under Indian pharmacopoeia. Now we come to the last unit, that is good laboratory practices, good manufacturing practices, and good clinical practices. After studying this unit, you should be able to explain principles of good laboratory, good medical, and good clinical practices, understand the responsibilities of quality assurance program, explain the functions of good laboratory practices and good laboratory practices inspectors, describe the features of aseptic area in GMP, discuss the role of personnel in GMP, explain the importance of GCP, that is clinical practices, understand different phases of clinical trials, describe the importance of declaration of Helsinki and Schedule Y, and understand institutional review board, independent ethics committee. First, we come to good laboratory practices. What is good laboratory practices? A number of countries require manufacturers of pharmaceuticals, veterinary drugs, pesticides, cosmetics, processed food products, feed additives, industrial chemicals, etc., to establish that use of these products do not pose any hazards to human health and environment. Non-hazardous nature needs to be established through studies and data, which will be examined by the regulatory authorities of the concerned countries. Good laboratory practices is a system which has been evolved by Organizations for Economic Cooperation and Development (OECD) using used for achieving the above goals. To avoid different schemes of implementation that could impede international trade in chemicals, OECD member countries have pursued international harmonization of tests and methods and good laboratory practices. The National Good Laboratory Practices Program functions through an apex body which consists of the following members: a chairman, who is the Secretary, Department of Science and Technology or Ministry of Science and Technology of Government of India, then various members or secretaries of Department of Chemical and Petrochemicals, then Department of Agriculture and Cooperation, Department of Health, Ministry of Health and Family Welfare, then Drugs Controller General of India, Secretary of Department of Commerce, Secretary of Ministry of Environment and Forest, Secretary of Department of Fertilizers, Secretary of Department of Consumer Affairs, and Director General of CSIR and Secretary of DSIR, Head of National GLP Program of Government of India. Now, definition and scope. Good laboratory practices is a system of controls and management for laboratories and research organizations to ensure the consistency and reliability of results as outlined in the OECD principles of GLP and national regulations. Good laboratory practice embodies a set of principles that provides a framework within which the laboratory studies are planned, performed, monitored, recorded, reported, and archived. So, all the methods and all the phases of production of drugs comes under good laboratory practices. Then powers and functions of GLP authority. The main functions of GLP authorities are monitor the progress of program implementation, establish national GLP compliance monitoring system, grant GLP certification to the test facilities based on their compliance to OECD principles, suspend, withdraw, or terminate GLP certification from its certified test facilities, and may even inform relevant GLP compliance monitoring authorities should there be any need. Constitute such other technical committees or the working groups which it deems fit to complete a particular cause or need or activity. Approve the rules and the technical committee and working groups, ensure that national GLP compliance monitoring authority operates its system in accordance with current OECD council norms, maintain its international compatibility and mutual recognition, and lastly organize and conduct scheduled unscheduled inspections for its GLP certified laboratories. So there are various functions of GLP which we have discussed. The nature of national GLP program. The good laboratory practices certification is voluntary, which undertakes such studies either for its own purpose or for others will be eligible to seek GLP certification. It will establish and continually update a network of GLP certified laboratories. GLP certification is valid for a period of 3 years. Then GLP inspectors. The national GLP program has opted in its system to impanel as its inspectors the experts who are currently employed with government test facilities, organizations, and whose qualifications, experience, etc., are meeting those prescribed by the technical committee. The inspectors have various functions under GLP similar to the functions of GLP. But GLP principles and requirements, test facility organization and personnel. Then quality assurance program should be there, facilities to conduct the various tests should be there, apparatus, material and reagents, test systems, test and reference items, and standard operating procedures, performance of the study, reporting of study results, storage and retention of records and materials. These all come under GLP methods. Then good manufacturing practices. Good manufacturing practices is a term that is recognized worldwide for the control and management of manufacturing and quality control testing of food and pharmaceutical products. GMP takes the holistic approach of regulating the manufacturing process and laboratory testing during the process itself. Good manufacturing practices gives much significance for the documentation of every aspect of the process, activities, and operations involved with the drug and medical device manufacture. So not only during the laboratory preparations of the drugs, but during the manufacturing process of the drugs, GMP, that is good manufacturing practices, should be held. Locations and surrounding. The pharmaceutical manufacturing unit shall be situated in a clean and tidy environment, free from any type of contamination, including external environmental, including open sewage, drain, public lavatory, etc. The buildings for the manufacturing unit shall be designed, constructed, adapted, and maintained to suit the manufacturing operations so as to permit production of drugs under hygienic conditions and to avoid various hazards including entry of insects, pests or rodents and mix up between different categories of drugs and raw materials. Then water system. There shall be validated system for treatment of water drawn from own or any other source to render it potable in accordance with standards specified by the Bureau of Indian Standards or local municipality, as the case may be, so as to produce purified water conforming to pharmacopoeial specification. Purified water so produced shall only be used for operations except washing and cleaning operations where potable water may be used. Then warehousing areas shall be designed and adapted to ensure good storage conditions. They shall be clean, dry and maintained with acceptable temperature limits where special storage conditions are required, example like temperature and humidity. So warehousing should be clean and dry and maintained within temperature limits. Then production area. The production area shall be designed to allow the production in a scientific and professional manner. There shall be provision for attracting the risk of cross contamination. Self contained facilities shall be made available for the production of sensitive pharmaceutical products like penicillin or biological preparations with live microorganisms. Then, ancillary areas, rest and refreshment rooms shall be separate from other areas. These areas shall not lead directly to the manufacturing and storage areas. Facilities for changing, storing clothes, and for washing and toilet purposes shall be easily accessible and adequate for the number of users. Then quality control area. The design of the laboratory shall take into account the suitability of construction materials and ventilation. Separate air handling units and other requirements shall be provided for biological, microbiological, and radioisotopes testing area. Then personnel. Prior to employment, all personnel shall undergo medical examination, including eye examination, and shall be free from tuberculosis, skin and other communicable or contagious diseases. Thereafter, they should be medically examined periodically at least once a year. Records shall be maintained thereof, and the license shall provide the services of a qualified physician for assessing the health status of the personnel involved in different activities. Then raw materials. The license shall keep an inventory of all raw materials to be used at any stage of manufacture of drugs and maintain records as per Schedule U. All incoming materials shall be quarantined immediately after receipt or processing. All materials shall be stored under appropriate conditions and in an orderly fashion to permit batch segregation and stock rotation by a first in first expiry or first out principle. Equipment. Equipment shall be located, designed, constructed, adapted, and maintained to suit the operations to be carried out. The layout and design of the equipment shall aim to minimize the risk of errors and permit effective cleaning and maintenance in order to avoid cross contamination, buildup of dust or dirt, and in general any adverse effects on the quality of the products. Then documentation and records, which is very important. Documentation is an essential part of quality assurance system and as such shall be related to all aspects of good manufacturing practices. It aims is to define the specifications for all materials, method of manufacture and control to ensure that all personnel concerned with manufacture know the information necessary to decide whether or not to release a bath of drug for sale and to provide an audit trail that shall permit investigation of the history of any suspected defective batch. Then labels and other printed materials. Labels are absolutely necessary for identification of the drugs and their use. The printing shall be done in bright colors and in a legible manner. The label shall carry all prescribed details about the product. All containers and equipment shall bear appropriate labels. Different color coded tablets shall be used to indicate the status of a product, and printed packaging materials and products leaflets relating to different products shall be stored separately to avoid mix up chances. And quality assurance, this is a wide ranging concept concerning all matters that individually or collectively influence the quality of a product. It is totality of the arrangements made with the object of ensuring that products are of quality required for their intended use. The system of quality assurance appropriate to the manufacture of pharmaceutical products shall ensure that the pharmaceutical products are designed and developed in a way that takes account of the requirement of good manufacturing practices and other associated codes such as those of good laboratory practices and good clinical practices. Quality control system. Quality control shall be concerned with sampling, specifications, testing, documentation, release procedures which ensure that the necessary and relevant tests are actually carried and that the materials are not released for use nor products released for sale or supply until their quality has been judged to the satisfactory. It is not confined to laboratory operation but shall be involved in all decisions concerning the quality of the product. It shall be ensured that all quality control arrangements are effectively and reliably carried out. The department as a whole shall have the other duties such as establish, evaluate, validate, and implement all quality control procedures and methods. Then product containers and closures. All containers and closures intended for use shall comply with the pharmacopoeial requirements. Suitable validated test methods, sample sizes, specifications, cleaning procedure, and sterilization procedure, wherever indicated, shall be strictly followed to ensure that these are not reactive, additive, absorptive, or leach to an extent that significantly affects the quality or purity of the drug. No second hand or used containers and closures shall be used. Master formula records. There shall be a master formula records relating to the manufacturing procedures for each product and batch size to the uh, to be manufactured. These shall be prepared and endorsed by the head of production and quality control. The master formula shall include the name of the product, the patent or proprietary name of the product, name, quantity, and reference number of all the starting materials to be used, a statement of the expected final yield with acceptable limits, and a statement of processing location and principal equipment to be used, the methods or reference to the methods to be used for preparing the critical equipments, detailed stepwise processing instructions and the time taken for each step, the instructions for in process control with their limits, the requirements for storage conditions of the products, including the container labeling and special storage conditions where applicable, any special precautions to be observed, and packing details and specimen labels. Now, packing records. There shall be authorized packaging instructions for each product, pack size, and type. A batch packaging record shall be kept for each batch or part batch processed. It shall be based on the relevant parts of the packaging instructions and the method of preparations of such records, such to be designed to avoid transcription errors. Then product recalls. A prompt and effective product recall system of defective products shall be devised for timely information of all concerned stockists, wholesalers, suppliers up to the retail level within the shortest period. The license may make use of both print and electronic media in this regard. There shall be an established written procedure in the form of standard operating procedure for effective recall of products distributed by the licensee. Complaints and adverse reactions. All complaints thereof concerning product quality shall be carefully reviewed and recorded according to written procedures. Each complaint shall be investigated, evaluated by the designated personnel of the company, and records of investigation and remedial action taken thereof shall be maintained. Reports of serious adverse drug reactions resulting from the use of drug along with comments and documents shall be forthwith reported to the concerned licensing authority. Then specific requirements. Sterile products being very critical and sensitive in nature, a very high degree of precautions, prevention and preparations are needed. Apart from the general requirements of good manufacturing practices of premises and materials for pharmacological products, some additional requirements shall be complied with for the manufacture of sterile products, parenteral preparations, and sterile ophthalmic preparations. Then aseptic areas. The sterile items has to be manufactured in aseptic conditions to make them free from microorganisms. Extra care and precautions have to be taken in the design and construction of aseptic areas. Walls, floors, and ceiling should be impervious, non-shedding, non-flaking, and non-cracking. Flooring should be unbroken and provided with a curve both at the junction between the wall and the floor as well as the wall and the ceiling. Walls shall be flat and ledges and recesses shall be avoided. Ceiling should be solid and joints shall be sealed. Light fittings and air grills shall flush with the walls and not hanging from the ceiling so as to prevent contamination. Then garments. The garments required for use by personnel working in aseptic area shall be of special type. Outdoor clothing shall not be brought into the sterile areas. The garments shall be made of non-shedding and tight weave material. Cotton garments shall not be used. The garment shall shed virtually no fibers or particulate matter which can cause contamination of the drugs. The clothing and its quality shall be adapted to the process and the workplace and worn in such a way as to protect the product from the contamination. Then good clinical practices. Origin of the concept of good clinical practices. The history of good clinical practices statute traces back to one of the oldest enduring traditions in the history of medicine, the Hippocratic Oath. As a guiding ethical code, the Hippocrates Oath is primarily known for its edict to do not harm the patient. However, the complexities of modern medicine research necessitate a more elaborate set of norms and guidelines. A physician has to comply with some ethical and scientific responsibilities, such as obtaining informed consent or which disclosing risk while involved in the biomedical research. Definition of good clinical practices. Good clinical practices is a set of guidelines for biomedical studies which encompasses the design, conduct, termination, audit, analysis, reporting, and documentation of the studies involving human subjects. The fundamental tenet of good clinical practice is that in a research on man, the interest of science and society should never take precedence over considerations related to the well-being of the study subject. The principles of GCP are given below. Clinical trials should be conducted in accordance with the ethical principles that have the origin in Declaration of Helsinki and that are consistent with GCP and the applicable regulatory requirement. Before a trial is initiated, foreseeable risks and inconvenience shall be weighed against the anticipated benefit for the individual trial subject and society. A trial should be initiated and continued only if the anticipated benefits justify the risk, means benefits are more than the risk. The rights, safety, and well being of the trial subjects are the most important considerations and should prevail over interests of science and society. The available non clinical and clinical information on an investigational product should be adequate to support the proposed clinical trial. Clinical trials should be scientifically sound and described in a clear and detailed protocol. The trial should be conducted in compliance with the protocol that has been received prior institutional review board, independent ethics committee approval or favorable opinion. The medical care given to and medical decision made on behalf of subject should always be responsibility of a qualified physician or when appropriate of a qualified dentist. Each individual involved in conducting a trial should be qualified by education, training, and experience to perform his or her respective tasks. Freely given informed consent should be obtained from every subject prior to clinical trial participation. All clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. The confidentiality of the records that could identify subjects should be protected, respecting the privacy and confidentiality rules in accordance with the applicable regulatory requirements. Investigational products should be manufactured, handled, and stored in accordance with the applicable good manufacturing practices. They should be used in accordance with the approved protocol. Systems with procedures that assure the quality of every aspect of the trial should be implemented. Helsinki Declaration. The World Medical Association had developed the Declaration of Helsinki as a statement of ethical principles to provide guidance to the physicians and other participants in medical research involving human subjects. Medical research involving human subjects include research on identifiable human material or identifiable data. The declaration was originally adopted in June 1964 in Helsinki, Finland, and has since undergone many revisions and clarifications. Clinical trial or study. Any investigation in human subjects intended to discover or verify the clinical, pharmacological, or other pharmacodynamic effects of an investigational products and identify any adverse reactions to an investigational product and to study absorption, distribution, metabolism, and excretion of an investigational product with the object of ascertaining its safety and its efficacy. The terms clinical trial and clinical study are synonymous. Clinical trials on patients in different countries are approved and monitored by different regulatory agencies. In India, it is governed by Drug Controller General of India (DCGI) office through its subsidiary known as Central Drug Control Organization. In UK, it is monitored by the Medicine and Healthcare Products Regulatory Agency as advised by the Committee of Safety of Medicines, while in USA, the Food and Drug Administration (FDA) is the charge is in the charge of the clinical trials. Now, institutional review board or independent ethics committee. The institutional review board or institutional ethics committee consists of a reasonable number of members who collectively have the qualifications and experience to review and evaluate the science, medical aspects, and the ethics of the proposed trial. It is recommended that these should contain at least 5 members, at least one member whose primary area of interest is in a non scientific area, and at least one member who is independent of the institution or trial site. Investigator. Investigator is an individual who actually conducts a clinical investigation under whose immediate direction the drug is administered or dispensed to a subject. In the event of an investigation, in the event an investigation is conducted by a team of individuals, the investigator is responsible leader of the team. An investigator is responsible for the ensuring that an investigation is conducted according to the signed investigator statement, the investigational plan, and applicable regulations. Sponsor. Sponsor means a person who takes responsibility for initiation a clinical investigation. The sponsor may be an individual or pharmaceutical company, government agency, academic institution, private organization, or other organization which takes responsibility for the initiation, management, and or financing of a clinical trial. Then contract research organization organization. The Contract Research Organization (CRO) is an organization that allows or offers clients a wide range of pharmaceutical research services. A sponsor may transfer any or all of the sponsor's trial related duties and functions of a CRO, but the ultimate responsibility for the quality and integrity of the trial data always resides with the sponsor. The CRO should implement quality assurance and quality control. Now, monitoring. The purpose of trial monitoring are to verify that the rights and well-being of human subjects are protected. The reported trial data are accurate, complete, and verifiable from source documents, and the conduct of trial is in compliance with the currently approved protocol or amendments with good clinical practices and with applicable regulatory requirement. Now we come to the last part, that is Schedule Y and Drugs and Cosmetics Act. Schedule Y deals with regulations relating to clinical trials requirements for import, manufacture, and obtaining marketing approval for a new drug in India. The procedure for applying for marketing approval depends on the status of the new drug, which can be broadly classified into three categories: new drug substances discovered which are already approved, marketed in other countries; new drug substances discovered which are not approved, marketed in other countries; and new drug substances discovered in India. So there are three categories in this schedule: new drug substances discovered which are already approved or marketed in other countries, or new drug substances discovered which are not approved, marketed in other countries, and new drugs discovered only in India. So there are different phases of clinical trials: phase 1, phase 2, phase 3, phase 4, and accordingly the drugs are approved. So I have finished the course which I have been given. So I would like to thank you, Ignu, for giving me this opportunity, and thank you very much, sir. So I think we can leave now. Hello, sir. Hello. Hello.